So now it is my great pleasure to introduce the Dr. Prabhakar. And Dr. Prabhakar obtained his Bachelor of Technology in Electronics and Communications Engineering from the Indian Institute of Technology, Madras, and he received a PhD degree in Applied Physics from Stanford University. He was sole recipient of the 2001 American Physical Society PhD Thesis Award from Beam Physics. And following postdoctoral fellowships in mathematics at Stanford University and genomics at the Lawrence Berkeley National Laboratory, he joined the Genome Institute of Singapore to start his own research group. His lab uses single-cell assays, spatial omics, epigenomics, and novel algorithms for basic science and translation studies of disease processes. Major achievement includes the first single-cell transcriptomics analysis of colorectal cancer, the first large-scale screen for histone association QTLs, and many things. He co-founded Human Cell Atlas Asia and is currently Associate Director, Spatial and Single-Cell Systems. And he is head of genome analytics for Singapore's National Precision Medicine Program. So today, the title of his talk is Single-Cell Data Analysis, Asian Immune Diversity and Cancer Cell States. It should be very interesting talk. So Dr. Prabhakar, so if you are ready, please start.
Thank you very much for the wonderful introduction, and thank you to the organizers also for bringing me here. I hope you're not all tired of hearing long talks and at the end of this very exciting meeting. So today, I'm going to talk to you about single-cell data analytics, but first, let me zoom out a bit and tell you what the Human Cell Atlas is very briefly. So the Human Cell Atlas is an international consortium, a large number of countries, I think hundreds of researchers. And the goal of the Human Cell Atlas is to create a comprehensive reference map of all cell types in the human body and to map their properties as well. So you can think of it as a periodic table of human cells. Okay, so each element in this periodic table here is defined not by its number of protons, but its cell properties or cell type. Okay. So that's a Human Cell Atlas, and in this international consortium, we're acutely aware that current genomic atlases of all sorts are predominantly European. So you can see here that if you look at all GWAS studies, the red color indicates European origin samples for GWAS studies, and you can see that they account for the majority of GWAS studies. If you look at NOMAD, which is whole genome sequencing and exome sequencing, the red boxes here indicate European samples, and that's the majority of NOMAD samples. If you look at GTEx, which is genetics of human gene expression, again over here you'll see that around 85% of samples are of European origin. This is not for lack of trying, okay? It's just that I mean, all of these consortia, they're very much aware that you need international coverage for genomic atlases, and that's why you see diverse samples in there, but still they're a minority of the total. And that's primarily because all of this data is being generated, most of this data is being generated in the West. And so it will ultimately reflect the populations that are there in those countries. Okay. So genomic atlases are predominantly European, but diseases are global. Okay? And diseases are not just global, they also vary from country to country, or across geography, across ethnicity. I'll give you an example of that. If you look here, looking at coronary artery disease mortality rates on the y-axis as a function of cholesterol levels in your serum on the x-axis. And you can see here is that at constant cholesterol level, your ethnicity dramatically determines your risk of dying from coronary artery disease. Right? If you're an Asian Indian ethnicity, your risk of dying from that at the same cholesterol level is about 10-fold higher than if you're of Japanese ethnicity. Now, I'm saying ethnicity, this could be geography. Also, there's multifactorial, right? Could be genetics, environment, some combination. But clearly, diseases are not one thing, okay? And disease biology is not one thing. It varies across populations. And if you look at diabetes, it's just another example, complex disease. Again, diabetes prevalence varies by ethnicity, varies by geography, and really we want to understand what is the genetics and environmental factors in that. So global genomic medicine clearly needs global genomics, right? It cannot be only Western genomics. And that's where Human Cell Atlas Asia comes in. I told you about Human Cell Atlas. Human Cell Atlas Asia is the Asian arm of this international consortium. And it's very, very important for covering diversity or genomic diversity in the world, because 60% of the world's population lives in Asia. We have the two most populous countries, China and India. And across Asia, we have tremendous genetic, ethnic, cultural, environmental, socioeconomic diversity. And if you don't capture that in a genomic atlas, then you cannot really you properly use the genomic atlas for genomic medicine. So the flagship program of Human Cell Atlas Asia is AIDA, the Asian Immune Diversity Atlas. AIDA is a consortium covering 5 countries. So Singapore is the leftmost, and luckily in Singapore we have a multi-ethnic population. So in Singapore itself, in one lab, we can profile Chinese, Malay, and Indian ethnicity individuals. And then my AIDA collaborator, Jay Shin at RIKEN, is driving the Japan arm of the study on Japanese population. Woong Young Park in Samsung Genome Institute in Seoul is driving the South Korean arm. Partha Majumdar driving the Indian arm of this study, and Vardam Charan Sawan driving the Thailand arm of the study. So 5 countries, 6 population groups. And what we want to do across all of this diversity is generate a single cell atlas of healthy Asian immune cell states using single cell omics. We'll take that data, look for differences both within each Asian population, within each of these population groups, and across populations. And that's all healthy samples. So this is basic biology, right? No one has really looked systematically to say, if I look at blood cells in this population in this part of the world and blood cells in that population in that part of the world, how different are they at the molecular level? It's a very basic question. There have been some limited microarray-based studies on this a while back, very small cohorts. But I think you really need single cell analytics, and you need a large scale to do this systematically. And so in AIDA, in our pilot scale, we'll profile 500 samples. Right? So we'll generate single cell RNAseq data from peripheral blood mononuclear cells from 500 individuals across these populations. In phase 2, we use this healthy reference to compare to Asian disease cohorts. But in phase 1 itself, we're very excited to answer this basic science question. How different are human beings from each other if you look at their blood? So I mentioned the country leads in AIDA. We're very excited also to have the larger AIDA consortium led by John Chambers and Don Chong in Singapore, and Ian Tan as well. And then Chung Cha Hon in RIKEN, Ario Kawakami as well. In India, NIBMG, it's Arindam Maitra and Ankita Chatterjee, and in Mahidol University, Thailand, Ponpan alongside Varadon. So it's a large consortium already, but it's an open consortium, meaning we're happy and we welcome new cohorts and new countries into AIDA. So if you're interested in joining and mapping Asian diversity and bringing Asia into the Human Cell Atlas, please contact us. So a lot of work has gone into AIDA, and very happy to report that from a pilot data set across the first 3 countries, Japan, South Korea, and Singapore, we have minimal batch effect. So if you're not familiar with this kind of picture, I'll show you. So each dot here is a cell, and there's a 2-dimensional reduced dimensionality, so nonlinear dimensionality reduction method called UMAP. So you're projecting each cell as represented by its transcriptome, right? In its multidimensional transcriptome space, you're projecting it non-linearly into this 2-dimensional space. So the blue dots on top are the cells from the Japanese cohort, the green dots are from cells from Singapore cohort, the brownish dots are cells from South Korean cohort. And on the top left, you'll see all of them superimposed on each other. Now, the fear is that because these 3 data sets were generated in 3 different countries, in 3 different labs, collected by 3 different hospitals, actually more than 3 hospitals, that you will have profound technical variation. Okay, these dots, these cells will land in different places in gene expression space just because of technical biases in how this data were processed. But as you can see, the green dots, blue dots, and red dots, they lie right on top of each other. Okay, so they cover more or less the same regions of gene expression space, suggesting that whatever, if we have technical differences between these countries, they are minimal. And that's very reassuring. And that will help us compare across the countries. So AIDA is going to scale up to 500 samples. That's just a pilot. A lot of heavy artillery went into harmonizing those methods and also harmonizing the bioinformatics. So for the rest of my talk, I'm going to talk about bioinformatics methods. And for the first message I want to give you is that bioinformatics pipelines and single-cell data analysis are very complex. It's not that you just take your data, run one algorithm, and you get the result. This is what it actually looks like, and this is only actually phase 1 of the pipeline. Okay, so it's a long chain, and you have to get every link of this chain right, and you have to do every step of this pipeline to the highest quality if you want to do this industrial scale single-cell analysis across multiple countries. What do I mean by that? So you start with the raw data on the left, and this is next generation sequencing data, single-cell transcriptome data. These are just raw reads from the sequencer. And then you run an algorithm called Cell Ranger, which turns this raw data into a gene cell matrix, where each row in this matrix is a gene, and each column is a cell. Okay, so each column here is a single cell transcriptome, and you've got hundreds of thousands of cells. We hope to cross 1 million cells in AIDA. But this is just raw data. A lot of these cells are really low quality. You want to throw them away. Okay? You don't want to keep them. And so you do your QC, you discard things that are clearly not cells, and then you get your filtered gene cell matrix. But then you realize that there are some doublets. Okay, you think this single cell data, actually sometimes you've got 2 cells in 1 droplet, and so that's an artifact. And also, because we do this on a large scale, we pool our samples, we need to demultiplex them again. So there's some algorithms we use to demultiplex the samples and remove doublets until finally we have only single cells in our data matched to donors. Okay, we know which donor each cell came from. Then we do RCA2 for major cell type clustering. RCA is a supervised clustering algorithm that I'll describe soon, to identify the major cell types in the data. And then for each major cell type, we do quality control. So this red box, so each dot here is a cell again in QC space. And the red box shows you which are the cells that we think are of acceptable quality. All cells outside this red box, we throw away as low quality. And now you can see here that this is the zoomed in red box is our final single cell data set after QC, after demultiplexing and so on. So we cleaned it up. Then we use RCA2 to identify major cell types again on the cleaned up data set. And for each major cell type, we do feature selection, identify marker genes, and then identify cell types with some explicit batch correction as well. Very long pipeline, and at the end of it, we just have each cell annotated to a cell subtype. And then there's a lot of downstream analysis you want to do as well. So I'll talk about some components, the key components of this pipeline. But first, I want to talk about a component, the first component, which is often neglected in single cell science. And that is QC, filtering the cells, discarding the low quality cells. And QC is very important. Okay, so you can imagine in Singapore is very real now and all over the world. we are filtering the air that we breathe, and we're filtering the air that we exhale to get rid of harmful substances that we do not want in the air. Okay, so filters are very important. They protect you from unwanted substances. But you have to use filters consistently. Okay, you have to filter all of your data. You can't leave out anything. And you have to use your filters the right way. Okay, and improper use of filters, again, achieves nothing. And that's why QC has to be done consistently and it has to be done the right way. So what does single cell QC look like? I'll give you an example of this, okay? If you do the QC the wrong way, what happens? So this is cells in QC space again. X-axis is the percent of mitochondrial reads. That's a measure of how damaged your cell is, okay? So if you're further to the right, your cell is more damaged. Y-axis is the sensitivity, meaning how many genes you detect in the cell. And that is, you can say, the sensitivity of your data. So Y-axis is sensitivity, X-axis is damage. So you want to be on the left side of the X-axis, low damage. And you want to be on the high side of the Y-axis, so high sensitivity. Okay. But this red box here is your conventional QC cutoffs for cancer epithelial cells. Okay. And you can see here, this in the red circle over here, you have a large number of cells that are supposedly undamaged, but actually low sensitivity, meaning you're detecting very few genes in those cells. And that is really the majority of your supposed cancer epithelial cells. And I'll just give you the punchline. These are not epithelial cells. These are just debris or nuclei or fragments of epithelial cells. Okay. The good epithelial cells are actually up here on top. But your conventional QC is very lenient and you include this debris as well. So what happens when you try to cluster your cells, again here each dot is a cell, you get this blurry jumble of cells. And it doesn't look like what you expect. Okay, what do you expect? So now the cells are colored by patient in colorectal cancer. You expect each patient's cells look different in transcriptome space. And you can see here the colors slightly separate out, the patients slightly separate out, but not very well. They are still mixed together, which is non-biological. But if you do stricter QC, now you see this red box on the left has gotten smaller. We're looking at only the good quality cells. We're throwing away the cells in the bottom left-hand corner, for example. Now it looks more beautiful, and the patient-specific cells separate out much more cleanly and cluster much better. So strict QC is very important if you really want to understand the biology of your samples. Okay. And here's another example. This is actually COVID-19 peripheral blood patient single-cell RNAseq data that we generated in Singapore. There's this weird cell state you see here, these orange cells, that turns out actually they are doublets and debris. Okay. If you don't remove them, then you think that T cells over here have a continuous variation with, I think, B cells over here and myeloid cells over here, which is actually not true, right? The T cells and myeloid cells, they're very far apart in gene expression space. They don't have this orange bridge connecting them. They don't have any intermediate states. These are actually low quality cells that still make it through conventional QC, but then we do a stricter QC, and now you see here it's much cleaner. Okay, the T cells are over here, B cells are over here, myeloid cells are over here. They're very far apart, very cleanly clustered. And so, in fact, that's what we use for all our primary cell data analysis. We do strict QC, and also we use variable cutoffs, because you can see here on the y-axis that different cell types have different QC, different locations in QC parameter space. You cannot use the same QC for all cell types. So this helps us a lot in all of our single cell, COVID, cancer single cell data analytics, and that's QC. I mentioned clustering. That is another major challenge in single cell data analysis. And why do I say that? So this is an example. So this is an overview of how people do single cell clustering currently. Okay. And what do I mean by single cell clustering? So you put these single cells in gene expression space, each dot is a cell again, and you say, I'll partition my space by similarity. I'll take all my cells and say if these red cells on the top left, for example, they're similar to close to each other in expression space. I'll say they form a cluster. And so they must be one cell type. These blue cells on the top right, it must be a different cell type. And so I imagine that my clusters in expression space match biological cell types. And the way you form the cluster is by unsupervised clustering algorithms. K-means, hierarchical clustering, graph-based clustering. Some of you may be confused when I say unsupervised clustering, because actually clustering is supposedly a textbook example of unsupervised machine learning. So why do I need to say the word unsupervised when I say clustering? Well, as it turns out, there are supervised clustering methods, not very widely used, right? And so our suggestion is that in the single cell field, for sure, we actually need to use supervised clustering methods. So here's a, why do I say that? Here's a pitfall of unsupervised clustering. If you don't know the batches, you don't do explicit batch effect correction. The red cells are from one technology and the blue cells are from a different technology. And you can see here the cells separate by technology. They separate by technical bias when you do unsupervised clustering. You want the red cells and the blue cells to lie right on top of each other, and they don't over here. So profound batch effects, when you do unsupervised clustering, there are some formal methods to address these batch effects, but then they confound, if batch and biology are confounded, then those methods are not very reliable. So what I'll show you is that supervised clustering automatically takes care of batch effects for the major cell types. What do I mean by supervised clustering? So here's the general paradigm. On the left, you see your original data set. Each row is a gene. Each column is a cell type and made up of many cells. And yellow indicates high expression, purple indicates low expression. So you can see here these different cell types have different gene expression vectors, but I don't want to cluster them in this original genes versus cells space. I want to, so this is the space here. So each dot is a cell, each axis is a gene. I don't want to cluster in this space. So all supervised clustering methods cluster in this space or a reduced dimensionality version of the space. Sorry, all unsupervised methods. I want to cluster in a different space of cell types rather than genes, okay? Again, each dot is a cell. Now each axis is cell type, and the coordinate is how similar is this blue cell to cell type 1, how similar is it the blue cell to cell type 2, how similar is it to cell type 3, and those are the coordinates in this cell type space. And that's what RCA does, right? It projects your cells into cell type or reference cell type space and clusters in that space. Now you see here the same peripheral blood, single cell RNAseq data set looks very, very crisp in this when it's projected into the cell type space. Each row is a cell type, each column is a cell, and you can see very clean boxes in this cell versus cell type projection space. And each rectangular box here is a clear cell type. Okay, so these light blue cells are one cell type. They're very similar to monocytes and non-classical monocytes, dendritic cells. So these are myeloid cells. These dark blue cells here are one T cell subtype. These maroon cells here are different T cell subtype. Then this light green cells are B cell, NK cells, and now we get to B cells with different subtypes, plasmacytoid, dendritic cells, and so on. You get very, very crisp and unambiguous clustering in your projected reference space when you do supervised clustering. And I'll show you that this is the real value. On the right-hand side, again, is the same figure of unsupervised clustering where the cells separate out by batch. Left side, same data. You can see the red cells and blue cells are right on top of each other. So this is supervised clustering in the reference projection space, and the batch effects, the cells are right on top of each other. So these are the T cells, these are the myeloid cells, and if I remember correctly, these are the B cells. So this is why we use RCA for all primary sample and disease sample clustering, and we use supervised clustering for that. Now, before you do clustering, right, you need to do feature selection. What is feature selection? Feature selection is essentially marker gene selection. Which genes are you going to use for your single cell clustering? So if you look at this figure over here, each row is a gene. And you can see here each gene looks slightly different, right, across these cell types. So all of these genes are useful for clustering. because they can separate the cell types from each other. But this is after you've done your feature selection. Before you do your feature selection, there are many rows in this gene expression matrix that will just look black. Okay, they have more or less the same expression across all cell types. Those are not useful. They're just adding noise to your clustering, right? So how do you do your feature selection and reduce your gene set to just these marker genes that tell you that are different between the cell types? That is the single cell feature selection problem. And the way we went about it is we said, let's use some intuition here. Okay. So again, here's another example. Each row is a marker gene. Each column is one cell type. And you can see these are beautiful marker genes, okay, almost black and white. They express in virtually only one cell type. Now, what are the properties of these? If we could magically know these marker genes in advance, we would do very good clustering, right? If your marker genes are so good, the clusters will be very crisp. How do we find these? Now, on the right, you see a property of these marker genes, which is the gene-gene correlation matrix. Now, here, rows are genes, columns are also genes, and the color indicates how well gene I is correlated to gene J across the cells. Okay, you can see here that the gene-gene correlation matrix separates out into these nice rectangles, squares actually, along the diagonal, which means that markers of the first cell type correlate with other markers of the first cell type, markers of the second cell type correlate with other markers of the second cell type, and so on. Now, on the right-hand side, you'll see genes that are not cell type markers. They correlate with themselves just along the diagonal and nothing else. They don't correlate with each other. So that's it. Use gene-gene correlations to say which are the useful genes for clustering, which are these genes, and which are the useless genes for clustering, those genes on the right. And it's more complicated than that, but this gene-gene correlation matrix is our first step. Okay, what do we do after that? So here's a gene-gene correlation matrix for one data set. We don't want to choose all the genes in this correlation matrix, okay? Because as you can see, they're redundant, right? We just want to choose representative genes on this correlation matrix. So the first representative gene we choose is this gene in red over here, and we regress it out from the gene-gene correlation matrix, and it accounts for a lot of variation. Then you see there's still some variation here in the gene-gene correlation matrix, so we regress this gene out, and we regress the next gene out, and so on. And we keep on regressing genes out until we've explained most of the variation in the gene-gene correlation matrix. What do I mean by that? So we just do a scree plot, okay? X-axis is how many genes we've regressed out. Y-axis is how much variance we've explained in a gene-gene correlation plot. We look at the elbow of this plot and we say we'll stop our regression here, okay? We'll take the first 20 genes and that's it. They account for most of the variation in the gene-gene correlation matrix. So that's the seed, that's the kernel of our feature set. And you can see here, these are these 20 genes in gene-gene correlation space. Okay, each dot here is a gene now. Now these 20 genes, they cover that gene expression space, but they're individually not clean enough. They're still a bit noisy to really do accurate clustering. Okay, so then we start adding neighbors to these genes. We add new red dots, which are similar to these original seed genes. And then we stop at some point, and that's our final feature set, okay, for clustering. So we take this final feature set, and that's our algorithm. There's some more tricks to that, which I don't have time to get into. But our clustering, our feature selection algorithm is called dubstepper, okay, determining the underlying basis using stepwise regression. So how well does dubstepper do this correlation-based feature selection? You always want to benchmark your methods exhaustively. First, we looked at an anecdote. We tried to find a rare cell type. Okay, so again, each dot is a cell in expression space. These blue cells are a rare cell type, a rare cluster, very few cells that were very successfully identified using dubstepper. Okay, you can see a very small number of cells. Only half of them were detected as belonging to this cell type in the original publication. But dubstepper was able to identify many more cells, I mean, twice as many cells, I think, in that cluster using more accurate feature selection. So this is an anecdote. On your right-hand side, you see more systematic benchmarking. So y-axis is a silhouette index. And it's a measure of how well cells of different types are separated in gene expression space. Okay? So if cells of different types are separated well by your feature selection, silhouette index will be higher. You'll be higher on the y-axis. X-axis is how many genes you use for clustering. Okay? And then the red curve is dub-stepper. The other curves are competing feature selection methods. You can see here that consistently, regardless how many feature genes you select, dub-stepper feature selection separates the cell types much better in gene expression space. And that's why for unsupervised clustering, we always use dub-stepper feature selection. Now, one unexpected bonus. So everything I've shown you so far is single cell gene expression, okay, single cell transcriptomics. But in emerging technology in single cell is single cell chromatin profiling, and the assay is called single cell ATAC-seq. People don't use feature selection in single cell ATAC-seq. You essentially choose every location in the genome as a feature, or every regulatory element in the genome as a feature. And as I've suggested earlier, that's obviously not optimal, right? You only want to use the regulatory elements or the genomic locations that are different between cell types. And if you do that kind of feature selection, then your clustering will be more accurate. But single cell ATAC-seq data are very sparse. At each regulatory element, typically you'll have only 0 or 1 reads, right? So your signal is almost binary. Maybe you'll have 0, 1, 2 or 0, 1, 1, 3. But it's not a very bad approximation to say single cell ATAC-seq data are binary. If so, your traditional feature selection methods that I showed earlier work even worse, right? Because they're based on a variation at one individual genomic locus. So we said, let's try, in fact, that's why generally people don't use feature selection when they analyze single cell ATAC-seq data. So we said, let's try dub-stepper feature selection on ATAC-seq data and see what happens. So on your left, you can see that if you use dub-stepper feature selection, you nicely recapitulate hematopoietic stem cell differentiation in this bone marrow data set. Hematopoietic stem cells going to the erythroid lineage, the granulocyte monocyte lineage, lymphoid lineage. But if you don't do feature selection, it's chaos. Hematopoietic stem cells split into two groups, and the lineage differentiation is just not very clear. Right? If you use traditional feature selection, again, you just see a blob. You don't see clear these three lineages separating out. So we're very excited that correlation-based feature selection is general. It can be used on transcriptomic data, it can be used on chromatin data, presumably it can be used on protein data as well. And so the combination of RCA and dub-stepper is what we use for clustering in all our single cell data sets. So many more algorithms that we use, but I want to quickly give you some flavor of what we do with all of these algorithms, all this data generation. So I'll show you colorectal cancer. Okay, what do we want to do here? It's a collaboration with Ian Tan, who is a medical oncologist in Singapore, and Moon-Young Park, who runs Samsung Genome Institute in Seoul, and Sabine Tejbar in Belgium, Leuven. So this is Singapore data, 270,000 cells from colorectal tumors. And we want to say, what are all the major cell types? So each color is one major cell type in colorectal tumors. And then what are their finer states? Okay, so we'll take these major cell types. If I remember correctly, these orange cells are fibroblasts. And then zoom in and say, what are the subtypes of fibroblasts? What are the subtypes of epithelial cells? And so on, using dub-stepper and unsupervised feature selection. So here are the fibroblast subtypes that we detected. So it's a two-stage pipeline. You take these fibroblasts that are detected using RCA, do unsupervised feature selection using dub-stepper, and then cluster them and say these are fibroblast subtypes. Now, intriguingly, we found that fibroblasts from the core of the tumor, fibroblasts from the invasive edge of the tumor, they inhabited different regions in gene expression space. You can see here, for example, they have some invasive specific regions, invasive specific fibroblast phenotypes. It's quite interesting to speculate that maybe these fibroblasts are driving the invasive properties of the cancer cells at the edge of the tumor. On the other hand, you have some core specific fibroblast states over here, and these may be the most stable, these may be the cells that are helping the tumor survive hypoxia and survive drug treatment. So these kinds of cell types are the cell states, I would say, are what we want to interrogate further functionally. Some other intriguing aspects of cell states in tumors. So light blue cells are cells from nearby normal, okay? The normal, non-cancerous, non-tumor tissue. And the red cells are fibroblasts from the tumor tissue. You can see here the light blue cells in the non-tumor tissue, they are very discretely different, okay? You have discretely different subtypes. In the tumor, it gets blurred out for some reason, and that's a very intriguing behavior. The fibroblasts, which are not cancer cells, they lose their differentiated identity. And then they occupy all kinds of regions in gene expression space that are intermediate between differentiated fibroblast states. And that's weird because that's the kind of behavior you associate with cancer cells, not with these stromal cells that are associated, that are in the tumor, but they're not cancer cells. In fact, endothelial cells also show this weird behavior. Light blue are endothelial cells in nearby normal, and red are the endothelial cells in tumors. You can see, again, this differentiated identity is smeared out, it becomes blurry, and you have a continuum of states. So again, these are the kinds of cell states we want to interrogate functionally in colorectal cancer. Another application of single-cell omics in cancer is a collaboration with Sin-Tiong Hong at Duke and also Charles Chua and Cheng-Hui Zhu, where we want to do something very directly translational. We want to say, okay, you do single-cell omics, so we have around 380,000 single-cell transcriptomes here from 3 groups of patients. At diagnosis, so this is bone marrow single-cell data collected at diagnosis. But this is retrospective. So we knew that what happened to those patients over the succeeding years, over the years after they were diagnosed and after the bone marrow was collected. So these green patients did well, they responded to treatment. These blue patients didn't respond so well to treatment. And the red patients, they had treatment failure subsequently. And so if we could identify at diagnosis which patient is going to in future fall into the green, blue, or red categories, we could treat them accordingly. Right? And we could treat, for example, the red patients more aggressively if we knew that they were not going to respond well. Can single-cell omics give us some cell states that are unique to the green, blue, and red population so that we can actually use the cell states as diagnosed predictive markers, okay? Now, here's our data, and it's very exciting. So just look, so these green cells are controlled healthy bone marrow. Now, the red cells are from the red patients, blue cells from blue patients, orange cells from the worst prognosis patients. You can see here that there are differences, okay? So this group is all blue patients over here. This part of gene expression space is highly enriched for red patients, which had the best prognosis. This part of gene expression space highly enriched for orange patients, which had the worst prognosis. So now what we're doing is we're developing algorithms to take this data and find the 2 or 3 best genes that can tell us whether the cell lies over here in this region, on this region, or in this region. And so then those 2 or 3 genes can become predictive biomarkers of drug response in CML. And we hope that this can be a very general paradigm that can be applied to other methods as well. Okay, so I hope I've given you a flavor of the applications of all of these algorithms. I want to thank Chan Zuckerberg Initiative that funded our AIDA work and also A-Star funding. That's my lab. I've given the names of all the people participating in this work. I point out Florian, who's driving RCA2, and Bobby driving Dubstepper. All right, thank you.
Okay, thank you for your excellent talks. So now we can accept this couple of questions. There are 2 questions from audience in chat box, and shall I read that? So the first question is, so at some point of time, we may have to reach consensus for clustering between interactive gene groups from scRNA-seq and traditional RNA-seq data for some desired disease phenotypes. How do we come up with the precisions for this? So maybe I think you showed the supervised clustering for using the cell types, but so question is the how to deal with the disease phenotypes or something like that. So can you answer the question? So
Sure. I think there are many, when it's a question about the consensus between single-cell RNA-seq cell states, let's say, and traditional, which means bulk RNA-seq.
yeah.
There's, I think the word consensus can mean many things in this context. So one standard thing people do is, if your bulk RNA-seq data is from a heterogeneous cell population, what we're doing increasingly, we're using single-cell transcriptome types, you can say. So you take the transcriptome of each cell type that's there in the bulk, infer that from the single cell data, then use these single cell transcriptomes to deconvolve the bulk transcriptome to say how many of each of these cell types is there in your bulk transcriptome. Another thing you can do is, because bulk transcriptomics is cheaper, you can have a bulk transcriptomic data set from 1,000 cancer patients. Okay, so one of our collaborators actually has that. So you can take your single cell gene expression markers and say, okay, and create a summary statistic from those markers and say that summary statistic, how does it vary across patients in this 1,000 patient bulk RNA-seq cohort, or let's say TCGA, which also has bulk RNA-seq. So I think single cell RNA-seq and bulk RNA-seq are complementary, and bulk RNA-seq for a while will always be cheaper and more scalable than single cell RNA-seq. So you want to do a combination of both. You do single cell RNA-seq on, say, 100 individuals or 500 individuals, you do bulk RNA-seq on even more, and then you project the information from the single cell bioinformatics into the bulk RNA-seq bioinformatics.
Okay, thank you. And the second question is that what is the difference between unsupervised clustering, especially hierarchical clustering and the correlation analysis? So they share the same concept? I don't think so. Because the hierarchical clustering is based on the dissimilarity matrix between the genes, and you use the correlation analysis. So can you explain that kind of the difference?
Certainly. So the correlation analysis was used for feature selection, which comes before clustering. So we use a correlation analysis to say which genes do we use for clustering. You can think of each cell is a dot, is a point in 20,000 dimensional space, where each of those 20,000 dimensions is a gene. But most of those 20,000 dimensions are useless. They're just noise. Okay. What feature selection does essentially is say, reduces, throw away 19,000 of those 20,000 dimensions and keep only these 1,000 dimensions, and that's your starting point for clustering. Okay, so that's feature selection, and we use correlation-based feature selection to say which 1,000 dimensions or which 500 dimensions do we use for clustering. Once you've thrown away your useless dimensions and focused on only the useful dimensions, it's still a multidimensional space. You still want to cluster the cells. So then we reduce from that 500 dimensional space to, say, a 50 dimensional space of reference transcriptomes, okay, where in that 50 dimensional space, x-axis will be, say, naive B cells, the y-axis will be regulatory T cells, and the z-axis will be myeloid dendritic cells, and so on. Right, so you have now, that's the supervised part of your clustering. You've gone from 500 genes to, say, 50 or 100 reference transcriptomes. In that 50 or 100 dimensional space, you can do any clustering you want. Okay, now you just have cells versus some coordinates, right? Those coordinates in our case are reference transcriptomes. But each cell is a point in a 50-dimensional space. Then you can use hierarchical clustering, you can use graph-based clustering, you can use k-means clustering, you can really use any clustering algorithm you like. So this supervised clustering concept is about which space you do your clustering in. It's not really about how you do the partitions once you put the cells in that space.
So I think So there are a couple of questions also, but I think so the time is running up. So maybe some. Oh yeah, sorry. Yes, I see. Let me ask a quick last question. So, how can we utilize yeah what say so the last question is, how do we decide initially which feature selection is useful for any kind of data? So do you have some general principle or some so do you have some idea for useful for any kind of data in feature selection so context?
I think as you're suggesting, there isn't one strategy that fits all. Our philosophy always in bioinformatics is look at the nature of your data. I think there's a quote from the movie Silence of the Lambs, Hannibal Lecter, right? So he tells Clarice Starling, the FBI agent, of each thing, ask, what is this in itself? What is its innate nature? And I think he himself was quoting someone else. So that's what we say. What is the innate nature of your data? In our case, the innate nature of our marker genes is that they're correlated with each other. And so we use gene-gene correlations to do our feature selection. But in a different domain, different kind of data, the correlations between feature genes may not exist and may not even be relevant. So we always start with the intuition of what does the data look like. And I'll point out one thing. So it was surprising to us that in the single cell field, no one was using this innate nature of marker genes. So no one was using gene-gene correlations for feature selection, even though that is the central property of marker genes in single cell data. And I think that's what's really unique about that Hubstepper algorithm.
Okay, thank you. So I'm sorry, I cannot accept all of the questions because the time is now running up. So, and at